Caenorhabditis elegans. The C. elegans gonad, an extremely efficient reproductive system. Oviduct, spermatheca. Uterus, vulva, spermatotheca. Ovary, here you could see meiotic zone, mitotic zone. Distal tip cell, sperm, oocytes, embryos. Developmental biology portion. And adult, capable of egg laying, in vitro development, first cleavage, eggs laid at gastrula, 1 to 5-fold, 2-fold, 3-fold, ex utero development, arrest if no food, hatching, growing, starvation, high temperature, L2, predator, L3, L4, molt, young adult, L1, L2, L3, L4. L4 is the adult, young adult. Okay. Oocyte chromatin, anterior, PAR3 in context, MEX5. Sperm chromatin, posterior. Oocyte pronucleus, cytoplasmic flow, PAR3, PAR2, sperm pronucleus, CYK4, asters. CYK4, then PAR3, PAR2. AB, hypodermis, neurons, pharynx, other. EMS, MS, muscle, pharynx, neurons, somatic gonada, other intestine, muscle, hypodermis, neurons, germ cell, germline, muscle. Anterior posterior axis formation. Anterior posterior axis formation is determined by the position of sperm pronucleus. After the entry, centriole associated with male pronucleus push the nucleus to the nearest end of the oblong oocyte, posterior pole. The sperm centriole then organizes the microtubules in the egg and allow positioning of several maternal proteins in the future anterior posterior end. PAR3 become localized at the future anterior pole. PAR2 localized in the cortical cytoplasm closest to the posterior pole, and PAR1 in the future posterior pole. These PAR proteins align the mitotic spindle properly and partition the maternal transcription factors to their appropriate cells. One of the most important localized entity by PAR proteins is P granules that specifies the germ cells. It appears to be a cluster of translation regulators involving translation initiation factors, RNA helicase, poly-A polymerase. The P granules are localized to the posterior region during each division, so it is found only in P cell lineage and at last produce sperm and egg cells. Formation of dorsal ventral left and right axis. Dorsal and ventral axis is seen in the division of AB cell. As the cell divides, it becomes longer than the eggshell. This causes the cell to slide and result on anterior ABA cell and one posterior ABP cell. Squeezing also causes the ABP cell to take a position above the EMS cell. The ABP cell defines the future dorsal pole and EMS cell defines the ventral surface of the embryo. Left-right axis is specified at 12 cell stage, during which MS blastomeres contacts half of the granddaughters of the ABA cell, distinguishing right side from the left. Control of blastomere identity. Both conditional and autonomous specification if we separate the first two blastomeres P1 and AB. P1 cell develops autonomously without the presence of AB, but in isolation AB cell make a fraction of cell types, so its specification is conditional and needs the descendants of the P1 cell to interact with it. Then autonomous specification. P1 lineage show autonomous specification by internal cytoplasmic factors. P granules act as morphogenetic determinants in P cell. SKN1 PAL1, P1 proteins encode transcription factors that determine the fate of 4P1 derived somatic cells MSECD, SKN1 EMS blastomere. SKN1 activates MED transcription factors that determine the entire fate of EMS cell. PAL1 somatic descendants of P2 cell, PIE1 germ line fate. Conditional specification, it is seen in the endoderm cell lineage. After the fourth cell stage, EMS requires induction from its sister cell P2 for the formation of ECL. In the absence of induction, EMS blastomere form 2MS cell due to the autonomous specification from SKN1. The P2 cell also induces an induction to distinguish ABA from its sister cell ABP. ABA give rise to neurons, hypodermis and anterior pharynx cells. ABP makes only neurons and hypodermis only. If one reverses their position, the fates are also reversed. Fates are determined by their position in the embryo. Interaction between ABP and P2 is mediated by GLP1 protein on the ABP cell and ABX1 protein on the P2 cell. GLP1 is a member of the Notch protein family. ABX1 is a delta-like protein which acts as a ligand for GLP1 present in ABP. This ABX1 signal breaks the asymmetry between ABP and ABA since it stimulates the GLP1 protein solely on the ABP blastomere. Gastrulation, signal receptor, Wenger cell, ventral uterine precursor, vulvar formation, vulval precursor cells or VPCs.